Our next speaker is our abstract speaker, Stacy Wilder. She is currently the laboratory supervisor at the University of Missouri Research Reactor Center, and her research focuses on better understanding plant growth promoting bacteria. Her talk today is titled Meeting Agricultural Sustainability Through Plant Growth Promoting Bacteria, and examination of the mechanisms for improved host uptake of zinc nutrient in maize using functional mutants of Azospirillum brasilense. Let's welcome all for our talk. And again, if you have any questions, please type them in the chat.
Hi, everyone. My name is Stacy Wilder. I work at the University of Missouri Research Reactor in Rich Ferreri's group. And today I'm going to be talking to you about the improving host uptake of zinc using Azospirillum brasilense. Currently, the world's population is 7.8 billion. By 2050, it is estimated to be approximately 9.7 billion. You can see here the three projections depending on the amount of population growth. About 2/3 of this growth will be in Africa, where cereal production needs to increase 335% from 2010 levels to meet population demands. The global population depends on cereals to meet 43% of global caloric intake. The United States is the leader in corn production, with over a quarter of all corn coming from the United States. In order to meet growing population demands, agriculture needs to increase both quantity and quality of crop products. There we go. 9 million people die every year from hunger and hunger-related diseases. This equates to about 1,000 people an hour dying from undernutrition. For perspective, in January of 2021, 120 Americans were dying from COVID-19 every hour. We now have a solution to COVID-19, but there is no single solution to hunger, as it is a complex problem of calories, nutrients, and many other factors. Undernourishment is the single greatest threat to child survival worldwide and the underlying cause of nearly 1/3 of all child deaths. Many of these deaths can be attributed to chronic iron and zinc deficiencies. Is this due to insufficient quantities of iron and zinc in our environment? No. Iron and zinc are both highly abundant. The issue is chemical forms and availability for uptake and use in plant systems. Iron exists in soil in recalcitrant forms, such as silicates and oxides. Greater than 90% of zinc is in an insoluble form. Iron and zinc are important nutrients for healthy plant growth, as well as important nutrients in foods for healthy development. So how do we improve the availability of these essential elements in our cropping systems? One strategy is to try to leverage the soil microbiome to the advantage of plants growing in it. In a single teaspoon of soil, there can be more than 7 billion microorganisms. That is more than all the people on Earth. Just one of these microbes is Azospirillum brasilense, a gram-negative alpha proteobacterium known for plant growth promotion. For our research, we have investigated the effect of different functional mutants of this species, that have an effect on iron and zinc uptake and seed content in maize. We hope to use these mutants to examine mechanisms of plant microbe interactions. Azospirillum brasilense is perhaps the most studied of this class of bacteria and have been commercialized in some countries as an inoculant during seed sowing. This bacterium is known for fixing nitrogen and making certain plant hormones such as auxin. Azospirillum brasilense is a non-nodule producing bacteria. For our experiments, we inoculated roots at the seedling stage with 3 different strains of Azospirillum brasilense and grew the plants in our commercial growth chambers with clay pellets as a matrix to suspend the roots in nutrient media. We used a commercially available hybrid maize and set a 12-hour photo period with a daytime temperature of 25 C and a nighttime temperature of 20 C. Here's a graph that was produced in a 2015 collaboration with the Stacy Lab and the Ferrari Group, which at that time was at Brookhaven National Labs. This shows the rate of auxin biosynthesis and biological nitrogen fixation in the mutants we used in these experiments. You can see on the x-axis the actual values of nitrogen fixation found in these strains from direct measurement using radioactive nitrogen-13 labeled N2 gas. On the other axis, you have auxin biosynthesis, which was measured using carbon-11 labeled indol. These spontaneous mutants that we are using are FP10, a mutant that lacks active nitrogenase function and therefore cannot fix nitrogen. It can still produce auxin, albeit at a reduced level from wild type. IPDC, a mutant that is deficient in the IPDC gene, which is linked to biosynthesizing auxin via indol-3 pyruvic acid, the dominant auxin producing pathway. IPDC is capable of fixing nitrogen in quantities similar to the wild type and has a greatly diminished capacity to produce auxin. HM053, a constitutively expressed mutant of the NIF gene, HM053 has a NIF positive constitutively expressed mutant of the NIF gene that hyperfixes nitrogen and has a high rate of auxin production. I will be showing some figures from a work we published earlier this year in the ISME journal in which we investigated iron assimilation using these microbes. These figures will help compare our findings of iron and zinc. In these studies, we used radioactive isotopes of iron 59 and zinc 65 produced at MUR, the MU Research Reactor Center, to trace uptake of these elements in roots and shoots. These isotopes give off beta particles, which can be used for auto radiography imaging using phosphor plates, and gamma rays, which can be used for tissue counting. We did separate experiments in which we added radioactive iron 2 and iron 3 and divalent zinc to the water in which roots were suspended. We used a sodium iodide photomultiplier tube detector to visualize uptake of iron and zinc into the upper plant parts. After 3 hours, we removed the plants from the solution, separated the roots from the shoots, and weighed the tissues and used a well-type detector with a sodium iodide photomultiplier tube to quantify the amount of radioactivity in these tissues. Here are the results of this experiment. The top scatter plot shows allocation percentages of iron 2 on the y-axis and iron 3 on the x-axis. Allocation is calculated as the percent of tracer which moved up to the shoot divided by the total activity taken up by the plant. As you can see, we found HM053 had the greatest uptake of iron 2 and iron 3, followed by FP10, IPDC, and control. The results from HM053 inoculation were unexpected because strategy 2 grasses would not normally take up iron 2. For zinc, the only mutant strain to promote uptake of zinc was IPDC. The percent allocation from roots to shoots for zinc was much less than for iron over the same window of time. You can see here on the axis. Inoculation of roots by each bacteria was verified by drop plate assay culture to assure that the roots were colonized. These are the images that we obtained from placing our plants on, these are the images we obtained from placing our plants that were treated with radio tracers on phosphor plates. Images were not normalized for intensity, thus are not directly comparable beyond location of the tracer. The plants on the right had different exposure times from 16 hours to 3 days. The plants on the left had exposure times of 3 to 6 days to properly capture the radioactive signature onto the phosphor plates. On the right are 2 images for the plants inoculated with HM053 and suspended in radioactive iron 2 and iron 3. You can see very different uptake in the leaf tips versus throughout the leaf. Typically, grasses such as maize do not assimilate iron 2, but utilize a mechanism whereby they make and excrete compounds that chelate with the iron 3, making it available for the plant root to take up the iron. The image of the plant inoculated with the HM053 mutant demonstrates that the iron 2 uptake has been turned on and the bacteria are not transforming the iron 2 to iron 3. On the left are 3 plants that are inoculated with the corresponding bacteria and suspended in that were inoculated with the corresponding bacteria and suspended in the solution of zinc 65. Note the uptake of zinc in the coleoptile and the base of the stem region and elongation zones, especially in the IPDC plant. Very little of the zinc has made it past the lower stem. The iron 2 and zinc show very similar uptake patternings in the leaves, but not the roots. Next, we used XLStat to perform a principal component analysis of our findings. PCA can reveal the most important characteristics within the data by plotting clusters of samples based on their similarity for multiple variables. These PCA biplots show the amount of variance among samples with the vectors root to shoot allocation and root uptake. For zinc, IPDC had the most unique allocation patterns with data points clustered on this side. HM053 had the most unique for ferrous and ferric allocation with data points clustered on this side and this side. In the summers of 2018 and 2019, we grew corn in 2.7 gallon pots in our outdoor space. We placed 3 seeds into every pot, let them sprout, and then thinned the sprouts. We inoculated the roots by injecting actively growing cultures into the soil. The soil was periodically re-inoculated. After the growing season, we harvested our corn and evaluated the corn yield as number of kernels per cob. And as you can see, HM053 had significantly more yield every growing season. IPDC, the auxin deficient mutant, with enhanced zinc uptake did not improve crop yield. Iron may be more critical to crop performance, or there may be other factors at work. Next, we examined the iron and zinc content in harvested corn kernels using ion chromatography. Again, we can see that the HM053 and IPDC inoculations gave very different results. While IPDC actually decreased the amount of zinc in the kernel, the HM053 inoculation significantly increased the iron content. The increase is approximately 3-fold in iron 3 uptake with no change in the iron 2 content. These results for IPDC are intriguing. IPDC increased uptake into the stem region, but decreased the amount of zinc in seeds. What could be behind this? Zinc is required for the synthesis of tryptophan by activating the tryptophan synthase enzyme for conversion of indole-3 glycerol phosphate to indole and ultimately to tryptophan. However, it is highly dependent on the concentration of zinc. At low concentrations, the tryptophan synthase is activated, but at higher concentrations, it is inhibited. Is this what we are seeing at the base of the stem? Does the plant reserve zinc in the stem and release it to the upper shoots to keep the proper balance of tryptophan synthase for maximum activation? Further studies are necessary. I would like to thank all my lab mates shown here in December of 2019. Hopefully next December, we will get to take a normal picture for December. I would also like to thank our financial supporters, NIFA USDA, Missouri Department of Natural Resources, DOE, Bayer Crop Science, and MUR. I would also like to thank you for your kind attention.
Thank you, Stacy, for sharing your interesting results. I see we have a question in the chat from Leticia. She asks, with the current situation of the pandemic, is FAO's forecast of a population increase to 10 billion still valid?
That's a very good question, and I'm afraid I don't know the answer to that. I would presume that it may perhaps the lower forecast, but I don't know whether our population, whether the effect of the pandemic has had that great effect on our population, because our population is so big, actually.
Awesome. Well, there may be more questions that are coming up in the chat. I had a quick question. So you mentioned that for some of the plants that you grew outside, right? You inoculated like some some of the bacterias. Like, did you do that all outside? And was there any difference of when you look at the plants that are grown outside versus ones that were grown inside, like in the younger versus older plants?
So we did grow plants inside and outside. Typically, we do most of our research with plants grown inside and they're young. We don't usually grow plants outside very often. We just don't have the place for it. We did find there are differences. Some of the differences we haven't measured on the inside plants, for instance, like stem thickness. Let's see, I think we've also grown some for like leaf length, leaf thickness, definitely. And we do see differences with the HM053 bacteria. The plants just seem to be a lot more vigorous.
Mm-hmm. Great, thank you. We have one more question from Dr. Paula McSteen. She says, very cool results. Did you measure auxin in the shoot versus root of the maize plants treated with the IPDC mutant to follow on zinc data?
So if I think I heard the question, did we measure the auxin in the roots versus shoots? Mm-hmm. I don't recall. I would have to speak with Rich. I'm sure he knows it right off the top of his head. If you could unmute him, I'm sure he knows the answer to this. Rich, are you there? Can you help me out? Yeah. Could you repeat the second part of the question as well?
it's basically, did you measure auxin in the suit versus the maize mutant treated with the IPDC mutant to follow zinc data? So in the IPDC mutant, when you treated, was there any difference in like auxin in suit versus root? I think that's the question.
And that's a good question. And I can't remember, but I know Rich knows. If you can Rich be unmuted? Rich Ferrari.
Yeah, easier. All right, he should be unmuted now. Rich, are you there?
We aren't hearing you, Rich, if you were talking.
All right, I'm sure we can follow this up later.
Yes.
If you have any question. But yeah, thanks for sharing your research and for interesting results. And so now let's thank our speakers again. We'll take a short break for 10 minutes, and then we'll be back at 10:30 to hear from Dr. Sarah Lira. We'll see you all after the break. Thank you for attending.
Thank you.